now she's going to do a presentation studying Tetrodotoxin levels across the life history stages of eastern newts. And by the way, she is the 2024 recipient of the Sherman Minton Award, the Student Presentation Award. So we have a check for you, Suzie. It is not a really giant check. I mean, it's not a giant physical check. A check for $300.
Thank you so much.
Efforts, and we thank you for being our guest speaker.
Yeah, thank you.
Can you please give Suzie
Well, thank you guys so much for having me. As he said, I'm about to talk about an assessment of tetrodotoxin toxicity across the life history stages of the eastern newt. So that's a lot of information tossed at you and a lot of big scientific scary words. So we're going to break it down a little bit first. So to start out, the eastern newt, or Notophthalmus viridescens, is part of the family Salamandridae. There are 4 subspecies across the eastern United States. They live about 8 to 10 years in the wild, and they breed from autumn to summer, and they particularly thrive in little weedy, fishless farm ponds. You can find them in a lot of places where there have been cattle, because they like dig those holes, and then there will be newts there. So why are newts exciting? I'm sure you all are excited about newts. We're at the Hoosier Herpetological Society, but what makes them different than any other members of the family Salamandridae? So what makes newts so different is they have a slightly different life history than other salamanders. So the majority of your salamanders start out as an aquatic egg and then become an aquatic larvae, and then they have an adult stage, either on land or aquatic. But what makes the newts so different is that they start out as an egg that their mothers will lay on vegetation. They grow to this larval stage, your kind of typical salamander larvae with big, beautiful, fluffy gills. And then they go on to this bright orange F stage where their skin thickens from what it was when they were living in the water. They become bright orange and they move on to land. And they spend a few years on land, 1 to 3, sometimes more, just foraging. You can find them under leaf litter if you're looking for herps. They're very easy to spot, as you can imagine, being that bright orange. But then when they reach sexual maturity, they return to the pond that they lived in as a larvae. And they kind of go through this secondary transformation where their skin becomes kind of soft, not as granular anymore. They become green instead of bright orange, and they grow a couple inches in size. And that is the adult aquatic stage. And then occasionally there is a 5th stage where once they have bred in these pools, they move back onto land, their skin becomes kind of harder and thicker again, and they will be terrestrial adults. So that's the life history of the eastern newt, pretty important for this conversation. The other thing that makes newts pretty important and very interesting to me is tetrodotoxin. So tetrodotoxin, or TTX as I'm going to call it, because tetrodotoxin is way too much to say every time, is a neurotoxin that it's named after the family of pufferfish in which it was originally discovered, but it's found in a lot of other species as well. And so what tetrodotoxin does is it binds with the sodium channels in muscles and nerves, and then it will immobilize those muscles and those nerves and can lead to respiratory arrest or even death in anything trying to prey on something that has tetrodotoxin. So I've got some pictures of the molecular structure up here. Pretty fancy and complicated. But what is so interesting about TTX, especially in newts, is the co-evolutionary arms race that's going on between rough-skinned newts in the western United States and garter snakes. So, as I said, newts have TTX in them, in their skin, and these garter snakes began to evolve to withstand some of that TTX. And as they evolved to be able to withstand that TTX and eat those newts, the newts started to evolve to have more TTX. And then the garter snakes started to evolve to withstand more TTX. So we're currently in the middle of this co-evolutionary arms race in which they're kind of fighting for who's going to be able to withstand more, And that is really what kind of this study and learning about rough-skinned newts is what made us interested in eastern newts a little bit, because we have a lot of information on rough-skinned newts and all the other western newt species, and how much TTX they have in them, especially over their life histories. But we didn't really have any comprehensive information on eastern newt species levels of TTX. There was kind of spotty data about, they'll have a study about the larvae here and a study about Fs here, but never one comprehensive study where you just go from one survey location and study the species throughout their entire life history stages. So that was what inspired us. So we kind of asked the question, how does TTX change across newts' life history? So I'm going to talk a little bit about our methods, and then I'm going to dive a little bit more into that. So it was essentially a 4-step process. I wish it was that easy in practice. It looks so neat and tidy up there saying, oh, we collected, we processed and froze, we extracted, and we assayed. But particularly for collection, we knew that we needed to be obviously hitting all of the life history stages, adults, Fs, larvae, and eggs. And we knew that we needed to collect them throughout the various points of the year to get an accurate reading on the data. So I have a few pictures here of us hunting for some newts, got a little newt in my hand, and then some of our lab-reared larvae that we had. So, collection techniques. This is the stuff that I feel like you guys are going to be really excited about, the actual herping stuff. So we collected our adults, particularly with minnow traps, and a lot of that was done in the winter. And we would set a minnow trap out in the little ponds where we knew there were newts, and then we'd come back the next day and check it. When we were doing that, we really struggled to get the females because they kind of tend to be out more in the water. But we collected lots of males with minnow traps. We used seines to collect larvae and then to also collect some of the vegetation that we found the eggs on. We used dip nets to collect larvae, and then some good old-fashioned getting out in the woods and going herping looking for our Fs, which was a bit of a struggle. It took a long time to find our Fs. But yeah, I've got some pictures up here of us collecting some adults out of the water. A really rainy day where we were really hunting for some Fs and we did not find any that day. And then another F hunting picture. So that was how we gathered the information, our specimens that we were using for this project. And once we had them, it was somewhat of a complicated process to get them to where we could actually assess the levels of TTX in them. So once we had the specimens in hand, we would process and freeze them. So what processing looked like was we would, with the adults particularly, we'd capture them and then we would take a 3mm skin punch and we would freeze this 3mm skin punch, and then we would, with everything else, with our Fs, with our eggs, with our larvae, we would weigh, record, and each individual got a specific ID number, so we knew where we had gotten it, what day we had gotten it, how much it weighed, what developmental stage it was in, if there was anything unusual in particular about that specimen, and then everything went into our freezer, so we all had it in one concise location. So you can see there 2 specimens with the 3mm skin punches taken out of their back, and then what those little tiny 3mm skin punches look like laying on the table. They get lost very easily, and that's not what you want when you're trying to use them for data collection. So once we had gathered all of our specimens, it was about February of this year, we went through the extraction process. So we ground all of our samples in a buffer, and then those samples were heated to 100 degrees Celsius, centrifuged for 20 minutes, and then they were strained and centrifuged again. And those final samples looked nothing like newts. It was just little bits of liquid in the bottom of a microcentrifuge tube. And they were placed in those microcentrifuge tubes and refrozen for transport. So the final step of this process was actually conducted all the way in Bakersfield, California at California State University with Dr. Amber Stokes. She is a colleague of my advisor, Dr. Gall, and she invited me out there to process my samples and to assess actually how much TTX is in those samples. So I was lucky enough to get to travel out there, and what the process looks like when we were
Just remember, we have a pointer.
Yes. Oh, this feels very bad. Now I kind of want to point to everything. So when I got out there, what that looked like is we had to defrost our samples, and then we would place them in microliter plates and wash them, which sounds really simple and puts a vision in your mind of like, oh, they put them in the sink and they wash them. I wish it was that easy. It was a very stressful few days for me, particularly because I am definitely field-based, and this was way lab-based, and I was like, I don't know what's happening. This is a lot of scary equipment. But after we washed them, we would dilute them with antibodies and then wash them again, and then we would read the sample's absorbance rates every 5 minutes for 40 minutes, which that looks so easy, just 3 little bullet points, but it would take the better part of a day to do about 50 samples. So you can see some of our little, I've got my little pointer now. You can see the microcentrifuge tubes up there, and those were all open so we could extract the liquid at the bottom that actually had the TTX in it. So now that I've told you all about what the processes were for all of that, let's talk a little bit about my results. So, first off, kind of disappointing I have to say this, data incomplete. It was actually very exciting for us that the data was incomplete because, first of all, it was incomplete because we had so many samples that it was just impossible to run them with how much time I had out there. But then second of all, these samples were way more toxic than we thought they were going to be, and so we are going to have to run a lot of those again, and we just haven't had the opportunity to do that. But we do have a little bit of a sample of what that data is going to look like. So what it looks like over the life history stages is eggs have about 8.5 nanograms of TTX, so just little dot right there. The larvae have 5,000 nanograms of TTX, so huge jump up there. The transitional larvae, so when their gills are kind of starting to shrink, their skin's starting to thicken up for that F stage, have 7,500 nanograms of TTX. There is a little noticeable gap right here. We know from previous studies that the levels of TTX in the Fs is going to be just off the charts compared to everything else here. But as I said, stuff is a lot more toxic than we thought it was going to be, so we don't have an accurate reading on that quite yet, but I'm looking forward to it. And then finally, our adult stages have about 55,000 nanograms of TTX in them. So, this is what it looks like for the eastern newts, but I kind of, at the beginning, I was talking to you guys about western newts and that sort of thing. So, this is this is a comparison about the western newts versus the eastern newts. So, as I said, the TTX in the eastern newts climbs throughout their life. It peaks at the F stage and drops back again at the adult stage, but still very high. It grows pretty generally throughout their lives. And that matches previous findings. As I said, there's never been a comprehensive study before, but in, collecting just eggs or just larvae, we knew that this was about what it was going to be. So in western newts, the TTX decreases throughout the larval stage. So you can see here 0 weeks to 28 weeks, it drops pretty significantly. And then it increases again post-metamorphosis. So there's metamorphosis and then you can see it growing again. And western newts have much higher TTX levels in general as compared to eastern newts. And this information is actually very interesting. It might seem a little bit, oh, there's just some charts up there, like, what's she talking about right now? But the fact that the TTX levels climbs throughout the life in eastern newts, but then it kind of decreases and then climbs with western newts, kind of points us in the direction that maybe the species are getting their TTX from different locations. There's not been much research done on that, but it would be an interesting thing to look at to see if, one of them is bacterial, one of them is dietary related, or something like that. And speaking of continuing our research, we have a little discussion about projects that we could work on in the future related to this. So why does TTX increase between egg and larvae? Is that a dietary situation? Who could say? We're not sure. Where does the TTX come from at all? As I said, There are thoughts that it might come from different locations in eastern newts as compared to western newts, which would be really exciting considering that they're pretty closely related, but might have completely different methods of getting that TTX. Something we would be really interested to know is, does maternal TTX correspond with the toxicity of eggs? So we'd be really interested to know if there's like a particularly toxic female that lays a clutch of eggs. Are those eggs going to be significantly more toxic as compared to eggs laid by a pretty average toxicity female? And then something that we're kind of hoping to look at with this project is there a difference between the TTX levels of lab-reared versus wild-caught larvae? You know, if they're living in a place where dragonfly nymphs are chasing them down all the time, are they going to want to develop to be more toxic in order to defend themselves from those dragonfly nymphs? Or is there really no correlation between that? So I have some acknowledgements on here. These people put a lot of effort into going into the field with me and working in the lab with me, and I'm very appreciative to all of them. And then I also have one more additional slide. So I actually received the email like, hey, you've been selected for the Sherman Minton herpetology scholarship in April. And I was kind of hopeful that I was going to be able to come in. You know, it's been a long time since April. But I actually spent this summer as a wildlife technician in California working with frogs, and I figured who better to talk to about frogs than the Hoosier Herpetological Society. So I worked primarily with Sierra Nevada and foothill yellow-legged frogs. So we called them wrassies and rabos. It'd be like, you're going out on surveys for wrassies today. And so what that looked like is we would go out to survey sites where we knew that there were frogs or that we kind of anticipated that there would be frogs that had never been seen there before. While we were at those survey sites, we would capture the frogs, measure them, weigh them, just collect a lot of data on them in general. If they were above 38 centimeters, they would receive a PIT tag number. So there's a PIT tag reader right there with the little frog that's we would take their pictures with their chin spots and correspondence with their PIT tag numbers, so we could later look at the pictures and be like, oh, like this guy is this guy, and stuff like that. One of the coolest things that we got to do is I was working for the Plumas National Forest, and we partnered with the San Francisco Zoo. The San Francisco Zoo would come to locations that we knew were going to dry out before the eggs could metamorphose, and they would collect those eggs, take them to the zoo, raise them to be huge, like monster frogs. The biggest yellow-legged frogs that we were finding out there were all zoo frogs. And then they would bring those frogs up to us, and we would get to put them in backpacks and hike them up to these very scary alpine lakes, and we got to release them. So this is a picture of our release crew. So our frogs are in those containers, and we would eventually pack them into our packs, hike them up, and then half the frogs would be in soft release containers away from garter snakes, so they had a little bit of a fighting chance against the snakes, and then half of them would just get hard released, and we kind of babysat them for a few weeks. We would go camp at the sites that we had released them to make sure that they were doing all right, not getting eaten by too many snakes. And then we also would test previously released zoo frogs for chytrid. So the San Francisco Zoo is doing some work on trying to make the frogs a little bit more resistant to chytrid. So it was really helpful for us to swab these zoo frogs to see if they had like the fungus growing on their outsides and if it was affecting their health levels at all. So yeah, like I said, that's us with our release frogs. That's our little frog PIT tag number. Just a beautiful aesthetic picture of the frog. There's me putting a tracking tag on one of our zoo release frogs, and that's me chest deep in a beaver pond trying to find some frogs that were not there. But does anybody have any questions about newts or about yellow-legged frogs? Yeah.
The tagging frogs above 38 centimeters, like, does that mean like they were 38 centimeters tall, they were located in the water? Like,
It would be 38 centimeters from the toe to like in the spine, because the PIT tags could affect them if they were any smaller than that. So yeah. Yeah.
I know that some zoos were working with different chytrid treatments on And you said that they were trying to test them and see if are they pre-treating them with something before release?
I believe that they were, like, there was some kind of pre-treatment, like an exposure of some kind. I'm not 100% positive because I was really more on the end of like, oh, here's a frog, here. We'll take that swab and we'll send it to San Francisco and they'll do something with it. But we did get to see where they were like exposing the frogs, and there was some kind of, I'm really not that sure on the procedure, unfortunately. I wish I knew more, but being on the field side, I don't have that much information on that.
How'd you get rid of the garter snakes?
The garter snakes would have to be, if they were in our like soft release areas or our hard release areas, we would put them in our frog catching nets and then hike them down the mountain to the alpine lake at the bottom, where they could still get food, but just not our very important endangered frogs that we were trying very hard to protect. So it was an interesting process dealing with the snakes. There was actually one day when I was out on hitch, kind of babysitting my frogs, and I found a snake right near the water, and I scanned it with my pit tag scanner, and I was like, he ate a frog. I wasn't very nice of him, and I was gonna have to use my net, so I was like, oh, I'll just hike the snake, you know, a mile away, try and release it, and, you know, hopefully he won't come back. It's all I could do in that moment. And so I'm hiking him out there, and then the snake starts acting really weird, and I was like, what are you doing? I'm looking at the snake. It's just opening its mouth all weird. And I knew that garter snakes would sometimes like vomit their food up. He vomits it up, and then the frog's laying there. I'm like, well, that's really sad. I don't want the frog to be dead. At least if he's gonna eat the frog, he should at least digest it so it wasn't for nothing. And then the frog starts hopping. And I was like, oh my gosh. And so I picked the frog up and I ran back to the lake and I like put him in there and he swam away. You know, it's very possible that he died after that, but it was very heartwarming for me. So I called it the Jonah frog because it survived getting swallowed.
That's funny.
I like it.
Yeah.
I know that red-legged frogs and yellow-legged frogs aren't doing well in the wild, and how the numbers in the wild?
I think our yellow-legged frogs were kind of on the up and up, but I don't really have the data, especially since the field season is still ongoing in California, so.
Okay.
So I have a quick question. So I used to be a former professor at Hanover, so you know Daryl Carnes.
Yeah.
Anyways, now I'm at University of Indianapolis. Anyways, my question is, so I have a colleague at University of Indianapolis who does SEM, work with salamanders, skin salamanders, and for instance, he's worked with slimy salamanders. And I'm a microbiologist, so I don't know all these. But there's some sort of gland that actually, there's some sort of gland that secretes the sticky substance that slimy salamanders produce. Is that the same gland that secretes the tetrodotoxins? How's it actually being administered by these salamanders onto the skin?
It is the same gland, according to my professor right there who gave me the eyeball and nodded at me. He would know way more about that than I.
So amphibians have both mucus and poison glands, and the granular glands and the poison glands are distributed primarily on the dorsal surface. Mucus is pretty much everywhere. Mucus is really just to keep it moist. The slimy secretions, they're producing TTX. Any toxin that the amphibians are producing are going to come out of those granular glands. A bunch of people, some of my colleagues have done some work on TTX specifically in those glands, and it definitely comes from those same glands. So they're just different toxins, presumably different genetic mechanisms to produce those toxins.
And so you were saying that the toxins possibly are actually produced by microorganisms.
Yeah, TTX is a really frustrating situation. It's a very simple molecule. Molecularly, it's very small, but we really have no idea how it's produced and who's making it and where these animals are getting it. So it's found in like an insane diversity of species, pufferfish and some invertebrates, flatworms. One of my colleagues just described it in a leech. There's a crustacean with it. A lot of amphibians have it. Most of the members of the salamander the newts have it. But really we don't know how it's made. There's no molecular pathway. There's one that's been proposed for newts. A colleague of mine has proposed one, but again, it's a long way from really confirming that. The thing that's really, can you go up to your actual data with newts? The thing that's really interesting to me is how from egg to larvae to transitional larvae and then eft. If she was to show eft, it would be like up in the sky. It's like it'd be 550,000 would be the number for an eft, nanograms anyway. So the fact that climbs pretty steadily through that larval stage is very strange because TTX in western newts is found in granular glands. Larvae don't have granular glands. These larvae should not have granular glands. The skin has not transformed yet. And so why TTX is increasing, I can't answer, but I'm guessing that it's either symbiotic bacteria or it's dietary related. I would probably guess it's some type of bacteria. We think that marine animals get it from bacteria. Yeah, so like pufferfish, for example, we think there's some species of like vibrio bacteria that produce it, and we think that's maybe where they get it. Western newts, the numbers, like it's hard to even make a comparison. Western newts have millions of nanograms. Some populations are low. A western newt could kill, if we were to inject it, hundreds of people. If we were to eat it, it could kill 30 people, 30 to 50 people. So yeah, this is really strange and really exciting because it means these very closely related species could be getting their toxicity from totally separate routes. I don't know, it's just really cool, very interesting.
One more question for you too. I'm actually teaching immunology this semester, and so I was wondering, so tetrodotoxin, as you mentioned, is a very small molecule. It's hard for me to imagine there's actually an epitope that actually interact with an antibody being that small molecule.
As far as I know, there are no antibodies. Well, the antibody that you run the assay with, yeah, I suppose. But I'm sorry, I was thinking like a binding protein is what I was thinking. But yeah, I know very little about the ELISA assay. That's why I send Suzie to California to work with Amber. So, but yeah, they've been making that. She's been running that assay for about not quite 20 years. There's one company that produces it based out of Hawaii. So, and it works very well. We've got data for tons and tons of critters using it. It matches, so far everything she's run has matched HPLC data that we've run to compare to. So.
Thank you, Dr. Gall, for the assist. Yeah.
I may have misheard what you said. Did you say the frogs were 38 centimeters?
Yeah.
I mean 3.8? 38 centimeters is,
38 millimeters, sorry.
oh, okay,
Okay,
okay.
all right, that's why I was confused
Yeah.
as well. It says centimeters on the thing.
I was working some of that through my head. That's gonna be a very big frog.
I've been in school mode and not frog mode for a few weeks, so I think that just,
Okay. I just needed that cleared.
yeah, yeah.
Yes. I just want to ask you, do you know some clinical complications for that TTX?
Some, what complications?
Clinical application in medicine or?
I don't know too much about like any sort of applications with that. I'm kind of field side of things. I like to catch the stuff and I let the micro people figure out what to do with it once we have that information. I'm not sure of anything. Do you know anything?
It's used extensively in like muscle physiology research, nerve function research. I've heard, kind of through the grapevine that some folks are looking at it from a pain relief perspective. So it's completely non-addictive. You cannot be addicted to it. So it'd be a really good application for it. The problem is it is so incredibly toxic. I'm trying to think of comparison. Pufferfish have relatively low concentrations, and yet, that's why people eat fugu. Fugu is pufferfish liver, right? Is to get that intoxication. People die every year just from eating pufferfish liver. So very, very toxic. And I think that's the issue, is translating it into a pain relief application. And if it's injected into the bloodstream, it's 40 times more lethal than if it's ingested. So it's just very dangerous. So even working with it, you have to be really careful. If it splashes in your eyes or in your mouth, something like that, you get cut on your hand. You have to be very careful.
Thank you, thank you.
Just a comment, I think you two both kind of answered that question, how they get this toxin into their system, but I know that, you know, most all amphibians have some degree of toxins, and these newts are known to feed on the eggs of, you know, Jeffersons and spotted salamanders. I didn't know if maybe that could be a way they could get their start with this toxin, possibility.
Yeah, it's possible, you know, just something that somebody needs to investigate. Yeah, and that doesn't super qualify for why we found any toxicity in the eggs. So that would be like, you know, kind of related to our, if it's a particularly toxic mother, is that going to correspond to toxic eggs? But yeah, that'd be something to look into for sure.
What is that gap in the adult stage where you got the big thing and then you got a little gap and then you got another spacing?
That's where the Fs would be. Like I said, we don't have like specific F data from this project, just because they were very toxic, so they need to be run again. But as both I and Dr. Gall were talking about, like if this were to be here, it would just be through the roof. It would be way higher toxicity than the larval stage or than the adult stage. So we just kind of have to imagine a vertical bar there that just goes woo, but we have to use our imagination for that one since we don't have the data quite yet.
All the way up to the space station.
Yeah, exactly. Extremely toxic. Yeah.
And the rare terrestrial form that becomes an adult, is that more toxic like the F or is it more like the newt?
That's also data that we're kind of waiting on. We were not expecting to find the terrestrial stage when we were doing this project. I just happened to stumble upon it while we were there collecting larvae. I walked over a little hill and accidentally kicked a rock and was like, what the heck is this? I've never seen this guy before. And I took him home and was like, hey, Dr. Gall, what's up with these weird newts? And that was a very exciting moment for us. So we're really excited to get that research, that, you know, information back, but we don't have it quite yet. So
As far as I know, no one's tested that stage. It's a pretty rare stage. I've seen a few, but I think you found maybe 7,
yeah.
which is very unusual. So we're very excited to get it. But yeah, I can't answer that. So we just don't know yet, unfortunately.
We're excited to find out with you. Yeah.
And also for all these newts that you're saying, when you're taking that biopsy bunch out of them, what are you doing with the animal? Is the animal alive? It goes back to being alive, or is it dead, or?
Unfortunately, we can't put them back in the wild just out of fears of cross-contamination or anything. So they were mainly euthanized in the name of science. But the population that we removed them from was doing just fine. There are plenty of newts there. So, yeah.
You mentioned there was an evolutionary arms race between the garter snakes and the newts. Do you know how much of the TTX it takes to kill a garter snake?
Oh, I don't know that much about the western newts. Do you?
It depends on the population. So populations are, there's 3 different species of garter snakes. There's 4 different species of garter snakes. 3 different species of newts out west. So it's very complicated. Garter snakes range from central California all the way up into Canada. Across that range, there are populations that are super resistant. So resistant that we could not buy enough TTX to slow them down to 50% of their crawl speed, let alone kill them. It would cost $1 million just to slow them down to 50%, not even kill them. Effectively, they're immune. Their sodium channels have changed so dramatically, and that's how they're resistant, is the sodium channel, imagine a tube, is literally changing shape. They're changing the pore of that channel so that TTX is floating through the bloodstream and cannot bind to that pore. So they've changed that so much that you can't affect them with TTX, essentially. Other populations are low resistance, like very close to baseline. And that's a really big question, is what initiates, what jumpstarts a population into that arms race, and what maintains it over, we're probably talking tens of thousands of years evolutionarily, where other populations, which might be only 100 miles away, are basically baseline. And we don't really know what jumpstarts it. It could be interactions with invertebrates. So one of the cool things is egg toxicity out west in Western newts is correlated with a female. So the more toxic a female, the more toxic her eggs, the less toxic a female, the less toxic her eggs. So maybe invertebrate predation on the eggs, you know, if a female has highly toxic eggs, and then a caddisfly goes to eat them, and the caddisfly can't eat it because it's too toxic. Well, that might jumpstart the coevolutionary process with garter snakes and get them interacting on a selective level. So it's a good question. I can't answer all of it, but yeah, it's variable. Toxicity is variable, resistance is super variable across the geographic distribution. So.
I have a question. I was talking to her and I wondered if maybe you can answer my question. Little background, I have an inspection camera that I've used to find animals hiding in hollow logs and rock crevices.
Okay.
And in southern Illinois, I found a neonate cottonmouth water moccasin in a rock crevice, and there were 3 young newts in there with him, crawling on him. And ever since I saw that, and I got pictures.
The F stage, the orange?
No, well, they weren't Fs, they were newts.
Adult stage.
Yeah, I can show you the pictures.
Yeah, show me the pictures.
Well, I was wondering, you know, did the snake, wouldn't he have eaten those, or maybe they were too small, or maybe they didn't smell right?
I would think that he would eat them.
Maybe he tried once and didn't like it.
It's definitely possible. We know from all kinds of different studies that it doesn't take much toxin. So the predators basically, they get that toxin in the membranes in their mouth, and they can tell that something's not right, and they'll spit it out. And so, you know, 1 or 2 interactions with one of those toxic prey is enough for them to reject them. Even invertebrates, dragonfly larvae will shoot their lower jaw out, pull them back in, take 1 bite, and spit them out. So that's very possible. I have a student who worked with cottonmouths, and they would eat about everything, but I don't think she tested newts. So it's a good question. And I would think that would be perfect prey for the neonates. Would be either the F stage or the adult stage. So.
Okay, well, sometime today I'd like to show you those pictures.
Yeah.
Thank you.
Very cool. And this is ongoing? Is it an ongoing study?
We are currently awaiting some of that data still from California, but this portion is wrapped up, but there's definitely way more to explore under this umbrella. The specific life history study is complete, but we'd love to look at the larval stage issues, like is captive bred versus wild caught? Is there a significant difference there? We'd love to look at the maternal levels. So the life history portion is pretty well wrapped up, but we have only, sampled in 1 location as well. It'd be really interesting to go somewhere else that has different levels of predation in different stages and look at how the TTX levels vary there. So this one specific, small subset of the project is wrapped up, but there's a lot more to do.
I was just curious if you had results of the F stage TTX.
Not yet. Still awaiting that from California.
Yeah, is there a way we can see those results? That'd be kind of neat to see it.
Yeah,
How far they actually are.
we'd like to see it too, that's for sure. But we know about where it's going to be, but we didn't want to include that data since it wasn't, we didn't want to include it, our guesstimation since it's not from this study.
Sure. Thank you.
Yeah.
The yellow-legged frog, the one that the garter snake ate and then threw up,
Yeah.
do they have any kind of a toxin too or not?
I can't speak to that super scientifically. I can say that a few of my coworkers would sometimes, we handle frogs all day. If they would touch the frogs and then touch their lips, their lips would get a little numb and tingly. I'm not sure as to specifics, but I would say that there's something up with them. And they also get really foamy the more you handle them. So I don't have a lot of information on that, but that would be something interesting to look at, yeah.
Sure.
Okay. Well, thank you guys so much for having me. I really appreciate it.